Today we have the chance to open a window, and thanks also to the people that are following online. Please, I just will ask you to mute your microphone, and later when it will be the interaction, you can unmute and maybe take questions. Thank you. And we have the pleasure to have among us two colleagues from the University of Debrecen. Debrecen is a university, an important university of Hungary, and is a very nice city, is a big university with a lot of students that comes from other areas of the world, so is a network hall. And it is pretty close to the Tokai region where there are good wines, so there is a good viticulture. So you will have an overview of the university and of on what they do at the university, and we can start a kind of discussion about the similarities with what there is in your experience, and if you are curious about some particular aspects that they will stress, we will hear a general presentation from Professor Peter Sipos, that is Vice Dean. He's very important in the management of the university, and we have also the chance, and you will have an overview of the university, on the activities that are run at university, and maybe, although you are at second year of master degree, you can have some experience, and you can go for traveling, or you can also eventually think to some experience that is postgraduate. So for PhD, or also for a short time for a visit or so on. And with University of Debrecen, we have this double degree for the junior, the students of bachelor degree, so Scienze e Tecnologie Agrarie. And so I think that there are chances for improving interaction and the communication about the two university. So let's start with Professor Peter Sipos, that will provide us an overview on the university. If you want, Peter, you can. You can hear by at home. It is clear. Can you hear? It is. Can you hear at home? I hear somebody, but I'm confident that they can hear. Okay, very good, Peter. You can come here.
So, Professor Manasi, thank you very much for your kind introduction, and of course for the invitation to give us the opportunity to, on one hand, we promote our university, the activity of your university, and give a chance to the students to visit us and contribute in our teaching activity and our research activity. And on the other hand, that you and your group and University of Ancona was always open for collaboration, and in the scientific field. And in the scientific field, there is a good communication and common activity with some fields of the agriculture. First of all, I would like to show a brief introduction of the university, and after that I will continue with the faculty, which works on the field of agriculture and food sciences. So first, University of Debrecen is the second largest university of Hungary. Hungary is not a huge country. Our area is approximately 100,000 square kilometers, and the population is just below 10 million inhabitants. But there are some things that we are very proud of. One, you can see that this is the 14th safest country in the world. And on the other hand, if you see the aspects of the agriculture, around one half of the area of the country is arable land. So some kind of agricultural activity is done on every second square meter of Hungary. The climate is a mild continental climate. However, the changes in the nowadays climate situation tends to some increase in the average temperature. And we used to discuss that maybe later, not so much time later, we also can contribute in the production of the tropical fruits with the Italian universities. The capital of Hungary is Budapest. You can see it in the middle of this. Okay. You can see in the middle of this map in the upper part of the country. Debrecen is approximately 220 kilometers from the capital, so it can be passed by train within 2 hours. So we are very close to the heart of the country. I don't know, have you ever been in Hungary somewhere? If somebody goes to Hungary, there are 2 locations which are...
Not yet.
Because professor didn't told you too much about the Hungarian wine. Okay. So if anybody thinks to Hungary, first they think to Budapest because the evening life and the social activity is very, very famous in the capital of Hungary. But the second one is the Lake Balaton, that you can see in the middle western part of the town. It is a quite large lake in our country, and we have 22 wine regions. 22 wine regions as well. So Tokaj is the most famous one, as Professor told you in the introduction, but not so far away from us, there is another famous one, the Eger wine region. And we, in our activity, have a good collaboration with these wine production technologies. And as I heard, this has a huge importance in your studies as well. So it can be a starting point for the common interest. The city of Debrecen is the second largest city of the country. It is more than 650 years old. You will see soon that the university is approximately 500 years old. So not so much after the establishment of the town, the education has been started in Debrecen. So we have a long tradition in this. The population is approximately 200,000 inhabitants. However, there are a lot of industrial improvements in the town and the country side of the town. BMW created a car producing plant by Debrecen. There are some accumulator plants are also built in the near of the town. This provides us a lot of work in the evaluation and the monitoring of the environmental aspects of this production, but this means that the population increases continuously. This number is referred to last year. The plans that the population of Debrecen will shortly increase by 10 or 15% in the next few years. We have a good opportunity in the case of the agriculture, because you can see in the bottom of this picture that we are on the very, very east side of the country, but this is almost the heart of the main agricultural area of the country. Beside Hungary, there is the Great Plain region of Hungary, which provides a good opportunity to produce crops and different kind of other industrial plants. And to the north of us, there is a very good area for the fruit and vegetable processing. 1/3 part or more than 1/3 part of the fruits and vegetables in Hungary are produced into the north of Debrecen. So from the agricultural aspects, our town is almost in the middle of the half of the agricultural production in Hungary. The connection between the city and between the university is very, very strong. And the university is one of the most important attractive points of the city. And this is why the city provides a lot of social possibility and free time activities for everybody. For example, you can see on two sides a lot of forests. In the side of the Debrecen, just approximately 500 meters from the university, not kilometers, meters from the university, there is a great forest, so-called great forest, which is a huge forest area. And somehow the university is just built by this great forest. The second thing is the thermal bath. The region of Debrecen is very, very rich in thermal water sources, which can be used both in spa activities and medical activities and just free time activities. Earlier, I thought that the university has approximately 500 years tradition in Debrecen. It was established, the first predecessor was established in 1538 as the Academy of the Protestant Church in Debrecen, and later different types of educational fields are appeared here, the theoretic sciences, the law sciences, natural sciences, and so on. And more than 150 years ago, it has been decided that the agricultural education has to also be appeared in this region. So the higher level agricultural education has been started in 1868, but it got the university level only about 60 years ago in 1968. This is the time when the Agricultural University of Debrecen has been established. Earlier, it was an agricultural academy, and joined to the other universities in Debrecen, in the heart of Debrecen, with the Science University, the Medical University, the Engineering College, and so on. And in 2000, all the different universities, there were 3 universities and some colleges, Debrecen and near to Debrecen. These universities joined to each other and formed the University of Debrecen. So the birth date of University of Debrecen is 2000. However, the predecessor of this university is approximately 500 years old. Now this university tries to provide education on all fields of the science. We have 13 faculties, and the activities are covering the medical and health sciences, the agriculture, food and environmental sciences, the humanities, music. We have a very, very good music faculty in the university, the law, different types of social sciences, natural sciences, informatics, engineering, childcare and special needs education. We used to say that every type of educational field are covered by the University of Debrecen, except for the military sciences and the theology sciences. Now we have 9 campuses, 5 in Debrecen. Faculty of Agriculture has our own campus, not in the middle, but very close to the middle of Debrecen. And we also have places, 5 locations countryside. Currently, the university has 32,000 students, and approximately 25% of them is international students. And the university offers more than 80 English language study programs on graduate and postgraduate levels too. The university is very proud that we won the award that we can be a science university. This means that very active scientific researches are done in the university. The most hot areas of these researches are the medical sciences, the natural sciences, and the agricultural sciences. The university got an award of National Excellence in the Research in 2013. And 2 years ago, our faculty also won the prize that the excellence research that we have an excellence research infrastructure in the field of agriculture and food sciences. And last year, we also got a second award in this field on the area of environmental evaluations. The university has 24 doctoral schools covering these researches and try to collect all the young researchers who want to improve themselves in these fields. The results of these researches can be seen in the rankings. The Times Higher Education and the QS ranking is monitored in every year by the university. And in the THA ranking, we can see that the University of Debrecen is between the first 200 in the European higher education universities, and in the world scale rank, it is between the first 800 to 1,000. On the field of agriculture, when the agriculture sciences are separately monitored, we are somewhere between the 250 and 300 universities in the DHA ranking or QS ranking. I don't remember some ranking. We have, in this semester, more than 7,500 international students. As you can see, here are the countries where the students are come from. You can see Italy, it is involved in the first column. 17 students we have now from Italy. The main region which provides us new students is Asia. Approximately 50% of the total students, international students, come from Asia. Approximately 20% from Europe and 20% from Africa, and the remaining 5-6% coming from America, the South and North America, and Australia. I think later in the faculty it will be also shown, but yes, it is a slide how the different international students can be grouped by the faculties. 2% of them are in the Faculty of Agriculture, Food Science and Environmental Management, or faculty. This means 150 students in the agricultural science. The most international students are on the Faculty of Medicine, Faculty of Engineering, Informatics, and Faculty of Science and Technology. Before I continue with our faculty, I would like to show you a short video, because the University of Debrecen has a lot of promotional videos, because I can talk about how you can imagine Debrecen and how you can imagine the university. Video can tell much more about it. So I try to start it. Oh. If you can help me.
Yeah. We stop sharing just to show to the people that we say hello to the people that are following online.
Yes, yes.
Here is. Oh, I think it should be some. Oh, yes. There is sound.
Yes,
But not here. Online.
but it's only music.
Okay.
It's auto, but it is not important. University of Debrecen has a lot of cultural activities. So, for example, there is the U-Day, which is a starting party for the students on the second educational week in the stadium of the university. Approximately 20,000 students are joining to each other and enjoy themselves.
Yeah, very, very nice.
I will give the slides of the presentation to Professor Ronaldo, so he can share with you both with the slides and both with us. If you are interested in, you can see it with sound. So we are from the Faculty of Agriculture, Food Science and Environmental Management, such a long name for a faculty. And yesterday we met with, the day before yesterday, we met with the director of your faculty, and he told that your activity also covers the same area, the agronomy, the alimentary, and the ambiente, sorry. So we try to work on the same fields. Here is our main building, but a much better picture is an overview about our campus. This campus is approximately 1.5 kilometers from the center of Debrecen, so it is very close to the center, and it is about 25 to 30 hectares in that part of Debrecen. The main building, what you have seen in the previous slide, is this one. So you can imagine what is the kind of this very, very huge campus. In this campus, we have a lot of facilities what we can use in the practical part of the education. If we go upward, so here is more road where we can upload to the end of the campus. If we go down to this road, first on the left, we can find an aquaponic and aquaculture laboratory. After that, there is a small research station for horticulture, and there are an older and a younger greenhouses for the horticultural researches and evaluations. If we go down, you can see some trees. That trees is a small botanical garden, especially for visiting or seeing the birds of the region, and we can use it in the education of the nature conservation study programs. This is called the bird-friendly park of the campus. If we go down, you can see some parcels. These parcels are research areas for the horticultural experiments, and after that, the crops, Institute of Crop Science researches are also can be placed there. And at the end of this campus, you can see a white building. This is a small food processing plant. In this food processing plant, there are 6 smaller units. We have there a milling microplant, a bakery plant, a food and vegetable processing plant where we can focus on the production of dried fruits and vegetables, juice and concentrates from them, and extraction and freeze drying. And yes, and then by them there are also in the base level meat processing plant and the milk processing plant. So almost all the agricultural activities can be first taught in the teaching to the students and do research activities. On the right side on this road, we can see some other places. There is a research area for the plant protection institute, thank you very much. And at the end, just by the food processing plant, there is a station for the engineering sciences connecting to the agriculture. On the other hand, there are sport activities also available on the campus. We always used to say that sport is very important for the students. University of Debrecen has a connecting athletic group. It is the DEATS, the University of Debrecen Athletic Club, and more than 30 different types of sports can be do there. So back to again, what kind of practical capabilities we have in this campus. We have greenhouses, we have aquaponic and aquaculture research station, we have this small food processing plant. But if we want to further more the basic agronomical activities, we can go to the animal farm of the university. It is something we can see. So there is an animal farm for the University of Debrecen at Kismac, which is approximately 2 kilometers from the campus of the Faculty of Agronomy, and some poultry and horse and sheep, and sheep can be evaluated there. There is a horticulture farm around 3 kilometers from the agricultural campus, and there is a arable farm. It is 15 kilometers from Debrecen, but it is quite a large farm. It is larger than 200 hectares, and approximately 1/3 part is dedicated to organic farming, so students can practice the basic steps of the organic plant protection activities. Some basic data about the faculty. So, do you know what is the program?
Lavoro.
Okay.
No, not touch me.
Okay. But we here can see what I was talking about. So we have approximately 1,500 students. About 1/10 part of them is international students. In the field of agriculture, we have 3 doctoral schools, one for the crop production and horticulture, one for the animal sciences, and one for the food sciences. In the staff, we have 236 colleagues. Most of them are teaching, participate in the teaching activities. Here you can see the institutes. You can see the institutes, but we have the basic institutes with the different areas of the agronomy. We have institute for crop production, horticulture, land use and engineering and precision farming and plant protection. In the animal field, we have a very, very large institute for animal sciences. For food sciences, we have Institute for Food Science, Institute for Food Technology, and Institute for Nutrition. And we also have a Water and Environmental Management Institute, which works with the environmental technologies in practice. We have 8 bachelor and 8 master courses, and nowadays, and with bold and blue, you can see, if it can be seen, the study programs which are taught in English too. In BSc, we have the agricultural engineering study program and the food engineering. And the newest one is the precision agriculture bachelor program. On master courses, animal husbandry, crop production, environmental management, food safety and quality engineers, plant protection and water management engineering are taught in English. And what we are proud of in the context of Ancona, we have a double degree study program for the agriculture and engineer BSc with the Università Politecnica della. This means that the students who want to select this opportunity and who want to apply for this double degree program has to do 1 semester study mobility in the partner university. So for example, the Italian students can go with Erasmus mobility program to Debrecen and collecting 30 credits. And after that, they can go back to Italy and finish the studies here. And after a half semester practice, they also can get the Hungarian diploma as agricultural engineer from the University of Debrecen. Now we are working a similar program with the food and beverages management and marketing study program from Ancona, your study program, and we hope that in the next year we also can offer it to students of Ancona and the students of University of Debrecen too. On the third hand, we also working with the double degree study program with the University of South Dakota. More or less it is ready. It seems that next month it will be signed by both universities. It will be established on the field of precision agricultural engineering, BSc program. For the research, the main strategic fields of the faculty can be grouped to 4 focus points. One is the molecular agriculture, targeted to develop and improve the plant and animal original genetic resources. The Second one is the application and the use of these genetical resources. So the future farming is supposed to investigate and develop new, modern, and research and energy efficient horticultural, arable farming, and animal production technologies. When we have the agricultural products, we have to turn them into products, foods, and other type of products. So the third one is the bio-based industry, cover the fields of the further use of the agricultural products. And the fourth one, everything is done in the environment. So everything is connecting to the surrounding environment. So we have to take consider on the agro ecosystems around us. So what we are working in the research is we try to develop site and variety specific precision arable farming and horticulture technologies in the field of the plant processing part of agronomy. We also do it in the field of the animal husbandry and animal nutrition, and we working on development of safe and traceable food products and processing technologies for mankind. We try to work and improve the integration of agri-environment schemes, nature conservation, and renewable energy systems into the agriculture. And we also evaluate the development of energy saving and environmental friendly technologies to mitigate the negative effects of the climate change. So we think that these 5 research areas can cover that strategic fields, but we plan to met from the demands of from the agronomy nowadays. As I told in our area, so in the field of agriculture, we have 3 doctoral schools. The first one is working with the crop production and the horticultural part of the agronomy. I try to collect what are the main research topics. So if you think about a mobility to Debrecen and connecting with a short research mobility program to some of our activities, you can select from this one, for example. So the genetic and environmental effect of productions are under investigation. We also evaluate the plant stress mechanisms and the bioactive and abiotic stress factors. I won't read all of them. We try to cover all the connecting areas from the crops, focusing mainly on the cereals, legumes, and oil crops. The horticultural products focusing on the apple, sour cherry, grape, maybe these are the most important ones in our researches. And in the case of the vegetables, the potato, paprika, pepper, and some smaller vegetables are in the focus of these researches, but we are mainly focusing on the... Yes, the pepper is paprika in Hungarian, so I always used to say paprika when I speak in English, but it is a bit for me. In the case of animal...
You use for goulash?
Yes. For example.
It's not good. It's possible to buy at supermarket and you cook at home, so it's good.
You don't have to cook at home. You have to go to Debrecen or you have to go to Hungary and you have to taste it because that one is the original one. Okay, in the field of animal science, the most perspective areas are the evaluation of the genetic variations of both wild animals and not wild animals, and they focus with the small ruminants, monogastric animals, and from wild and not wild species from the poultries. And we are also evaluating the biodiversity and the production and environmental relations of the animal husbandry. The food science is our field, especially so in the Doctoral School of Food Science and Nutrition, we are focusing on technological improvements and analytical method developments. And by this or within this, there are some research programs dedicated to the evaluation of the possibilities of increasing the shelf life of the different type of fruits and vegetables and how the pre and post-harvest treatments can help this. The next presentation will somehow connecting to this area. we evaluate the nutritive effects of different types of raw materials, how the different extracts, the chemotherapy products. Chemotherapy is, I don't know if you know what is chemotherapy. Chemotherapy. Chemotherapy, when we use the buds of the plants, because buds are very, very active parts of the plant. This means that a lot of nutritionally active compounds can be found in this, and some of the colleagues are working how we can use them, what are the possible benefits of them, and after that identification, what are those components which can cause these beneficial effects for these extracts. We also work with different types of cereals and industrial plants in the researches connecting to the food sciences. Of course, we also evaluate how the byproducts and the waste from the industrial food processing can be used for further purposes. And what is also active and hot topic is the nanoparticles and nanodots, the science of them, because similarly to the buds, they are also very, very active components from the inorganic compounds. And we don't know what can be their risks and what can be exactly their benefits, but we know that they are there, these particles are there, and in some cases, the normal food processing technologies increase the concentration of them in the foodstuff, so we have to know more about these nanoparticles. The student life in the video, you can see some very rapid but impressive picture about the student life. At the down part, you can see a lot of flags. This is the U-Day, the official study year opening ceremony. It is a moment from then when the international students go in and show their countries. And there is a lot of other events for the international students dedicated mainly for the international students. For example, there is the International Food Day, where these students coming from more than 40 countries from all around the world can produce their own foods, for example, goulash, the Hungarians, and everybody can try, everybody can taste the national specialties of other countries. There are some special events connecting to the agriculture. And I think the most important and traditional one is the yellowing for the agricultural students. Yellowing is a word from the production of cereals, production of wheat. Then the wheat grain is developing. First, it is green. After it became more and more pale green, and after that it go to yellow. And yellowing is a process when the students from the freshmen go to the engineer, and just before the engineer, they are yellowing, and this is a 1 week long celebration to almost closing the studies. And
they are close to the yellow.
Yes, you are close to that. And during this week, they have a lot of social activities. For example, in traditional clothes, they go through to the city on carriages, coaches, carriages, and do some dances in the heart of Debrecen, in the main square of Debrecen, and in the front of the building of the main building of the University of Debrecen. So we have a lot of traditions on this field too. And I don't know where is the second video, because I'm trying to add a second video. I will send you a link. It is a 3.5 minute long video, which was created to present all the possible activities of our faculty from the study, starting from the study area of the faculty and ending with this yellowing and other social activities for the students. So we have different websites if you want to know more information, if you want to get more information, what I was trying to summarize in this 20 to 30 minutes. The first one is the edu.unidep.hu website, which is the international educational website of the university, and you can find all the study programs which offer the University of Debrecen to the international students, independently which faculty it is connecting to which field of science. And on the other hand, you also can find here a lot of scholarship opportunities. For example, I don't know if you know that in Hungary there is a scholarship option, Stipendium Hungaricum. It is announced for the countries which are outside from the European Union, and the full study programs on BSc, MSc, and PhD can be covered from this fund. And we also have another English website dedicated to our faculty, and this is also can be seen at mac.unidet.hu/en, where you can see all the graduate and undergraduate programs connecting to the agriculture. These links are also collected on the last slide. As I tell, it will be sent to professor and he can share with you. But if you have any question, you can ask me now or just after this meeting, and you can find me on email. So do not hesitate if you have any question.
Hey.
Thank you.
In your side, you are the young student. Here there are some manager of the association of International Association of Agriculture. You have connection with Hungary? No? There is a station of YAS, that is International Association of Agriculture related students in Debrecen.
Yes, we have.
Because
International, yes. Something.
International Students Union.
because
What do you mean?
if you want, you can tell what you are planning for the next month. Who of you wants to? Okay, come on.
No.
Yeah, come here. Yeah. But this can be a further networking opportunity, because here in Ancona we have the reference for this network for Italy. For there is the president is from Ancona, and they are from the board of this association. Please.
The association?
No, thanks.
Was born and our associates are international association.
Yeah, there is this international association with a list of links and nodes in the different universities, and they will organize in July in Ancona a meeting. Actually, it's not yet in Ancona, but it is in different countries. So it's from Switzerland.
1 week, 3 days in Ancona, 3 days in Napoli. And 1 hour.
And then they meet in different areas. You are really Nepal?
Yeah, last year.
Last year?
And this year in Ecuador.
Wow. So there is this international network, they support each other and they have this kind of exchange. And maybe if you have connection with your students or student associations and wants to join them in Ancona, this can be a further opportunity for interaction. And if there is, they are welcome. If there is not yet, they may create and bridge, because it is a worldwide association of students in agriculture. So you can just digit IAAS.
Yes.
And you can.
No, I know, because we are from the Hungarian side in University of Debrecen Faculty of Agriculture. There is the national representative of this association. So I know, but I don't know if he knows about this event in July. I will ask him, and I try to enhance the connection between the Hungarian and the Italian side. Thank you.
Thank you. Thank you. Thank you. You have any question? Any other question? Okay. And now we can move to Professor Caraffa, that will have a presentation on the research activities on grapevine with some innovation. We talked about biological control. How much biological control is desirable.
Yeah.
But you have to find. Okay. How much biological control is desirable. But we need biocontrol agent. Then, in their lab, they discovered some biocontrol agents. They formulated. Okay. Okay, go down. Green. Yes. It is open. Fine.
Yes.
Okay, go. So, trichoderma is a novel. So, I will just increase the size. So, trichoderma is a biocontrol agent. It has target toward different diseases. We talked already, and we will talk along the course. And along the course, we will make reference to this seminar. So what it is in this seminar is integrated part of the courses, because trichoderma is one of the better known biocontrol agents, and it can be used to target a list of disease of grapevine. And here maybe we can follow a route that leads from discovery to formulation, registration, application. So, Professor Caraffa, floor is yours.
Thank you, and thank you for the possibility to show our results about these trichoderma strains. But first of all, some introduction about the importance and why we started to work on it. So I guess you have heard about the importance of alternative methods we should have in agriculture because of the European Party, because of the European Union strategy, farm to fork strategy, because of the consumers' requirement to decrease the pesticide residues in the food and to decrease the chemical pollutant of the environment as well. And we have further problems with the chemicals. For example, there are more and more reports about the emergence of resistance among the pests. So fungicides you are applying will not restrict the growth of the pathogens. And some new research also highlighted if you repress the usage to an environmentally friendly concentration, they will not successful anymore. So they will not have a biocontrol effect, control effect anymore. Another, which is not very widely propagated, that some of the pesticides has a plant toxic effect as well. Some of them are known, but some other chemicals may also decrease, for example, the photosynthetic potential for up to 1 month to be recovered after the usage. So Because all of these reasons, there are increasing demands to get new and other alternative treatments. And when we are looking for these alternatives, there are some which focusing on disease protection, either with controlling the pathogen itself or with inducing the disease resistance within the plants. So those can be both within the group of biocontrol agents and biocontrol chemicals. And the crop plant effect, some of them also can induce the stress tolerance. Okay, so they are either focusing on the plants, some of them focusing on the pathogens, and you will see that usually there is a mixture. So if there is a biocontrol effect, usually they also have increasing the stress tolerance usually. So as Professor Romanazzi told in the introduction, trichodermas are very well known. They are within the Hypocreaceae or Hypocreaceae family, which are well known by their mycoparasitic activity on basidiomycetes fungi. However, trichodermas themselves, toward the anti-basidiomycetic activity, they also can repress the growth of actinomycetes. Okay, so the broader targets toward the fungi. And their mycoparasitism is known from a very long time. So the first report was by 1922 and mentioning that they may have a role in wood disease control is as early as 1928. So they are well known from a long time. And where can we find them, those trichodermas? Because they are parasiting the basidiomycetes, so the mushrooms. You can find them on the surface of the mushrooms, and they are very widespreadly, can be found in soils and in other habitats, but for us, the most important one that you can find on the surface of the plants and inside the plants. So it's less known that not only in the soils, but on and inside the plants, you may also find them. Regarding their nutritional requirements, how to grow them, it's very easy because they can utilize several nutrients and sources. They can be, for example, can be produced a large conidia on agro-industrial wastes as well, okay? So whatever you put them, they will degrade. It's no matter, and they will grow on it. How do they affect? They affect on the pathogen because they are producing secondary metabolites, not only soluble metabolites, which can diffuse in the agar plates, but also volatile ones, okay, as a gas. We also have some results on it, but we are working on it. They also producing cell wall degrading enzymes. You can see in this picture, they are coiling, trichoderma means hair because they are very thin as a hypha. They are coiling around the pathogens. The other arrow show you they have a haustorium development. They producing enzyme make a hole. And in the picture bottom, you can see simple degrade, okay, digest the pathogen, okay? So they are eating and get the energy and the nutrients from the pathogens. And moreover, the third mode of action, they can grow very rapidly. So for example, our strains, within 3 days, they can cover the whole Petri dish. And they occupy the space and consume the nutrients. So the pathogens will not have either space or nutrients to grow when they are not trying to degrade the plants. On the other hand, they also active on plants. There are several studies that they are able to induce both the systemic and the local defense of the plants, so they can increase the resistance toward the pathogens. And moreover, they have also by promoting activity. So they also can promotion plant growth. They can increase the photosynthetic activity, and they also help for the plants to get the nutrients, different ions, for example, iron. Okay, they can help them, and they also can help to concentrate or get high concentration of potassium, for example, in the tissues. And we also did some studies. I will show you they can help for the plants to overcome the drought tolerance, for example. Okay, so if there are not enough rain, with the help of the trichodermas living inside, they can tolerate better. So although we know a lot about these trichodermas, in 2021, there were not too much species on the market, at least which were registered as biocontrol agents, because it's very expensive. It takes at least 5 years for the registration, a minimum €1 million. The company has to provide for this registration process. And although there are several products on the market in Italy as well, only some strains are there, at least in the biocontrol products. And sometimes not even the real names are in the products. For example, usually they are still talking about Trichoderma harsianum rifai. It turned out to be a different species within the Harsianum clades, so a group of species. So we know that those are Trichoderma afroharsianum. I just introduce you because that was among the first ones which is used. It was isolated in the States as a T22, but you can find the same strain registered as KRIAG2. Okay, several synonyms, even the strains. Sometimes there are more problems. For example, these other species within the Harsianum clade was withdrawn from the market. Not because it was not effective, because of economic reasons. So it was not profitable enough for the company to retain it in the market. Okay, so the huge majorities, those were isolated from soil. There are two ones, two Atroviridae connected to plant. One was from the leaf surface of the plant, but it is not in the market, I think because of economic reason as well, anyway. And the other one, which is still very popular, isolated, not inside the plant, but from the bark, decaying bark of a hazelnut. Okay, so none of them were isolated from crops, horticultural crops which are produced in the field and from inside the plant, okay? So I would like to introduce you our strains, our endophytic trichoderma strains. They were isolated accidentally. So we participated in a COST project together with Professor Romanazzi, and we were about to detecting grapevine trunk disease pathogens. And those were, we were screening whether the healthy plants also infected by the pathogens, what about the ratio of this infection. And we realized that there were those green trichodermas which overgrowth the pathogens. So the pathogens were inside the plant, we isolated from inside, and the trichoderma was there and didn't let the pathogen to grow. So it didn't eliminate the pathogen from the host plant, but didn't let them to grow. So the symptoms, I don't know whether you have learned about these symptoms of grapevine tongue diseases. It's a very complex disease, so more than 100 fungal species connected to it, which can cause the symptoms. They can have chronic symptoms, trigger stripping of the leaves or partial dying or the whole death of the plant as an acute symptom. And the most important one, there are several disease complexes. One of the best known is ESCA, and maybe it's the most important in Italy. The Botryopsphera, dieback, very similar symptoms, except there are a brown streak just below the outer part, below the bark. All cases you can see decay in the woody parts, other only brown or brown decay and brown rot and white rot. And Eutipa, it's not very important nowadays in Hungary because there are not enough precipitation for them. So when we checked the origin of those trichoderma strains, they were isolated from asymptomatic plants. So the pathogen were there, but the plants were healthy without any symptoms. And those were from woody tissues because we were about to isolate fungi from the woody tissues. We used those techniques. So we did this work together, my former PhD student, Csilla Kovács. So we worked further on it. First, we precisely identified the strains. And we were surprised because there were a lot of articles about endophytic trichodermas, but just Trichoderma sp or Trichoderma which were identified based on only ITS sequences. You have learned about it. Internal transcribed spacer is the most important barcoding for fungi they are using, but it's not enough for precisely identify the species of the trichodermas. So we used further markers, sequence markers for sequencing, TF1, translation elongation factor. And we could identify the species, and those were within Hertzianum, Longibrachiatum, and Viridic clade. So Longibrachiatum clade, several human pathogens were connected to it. And we also realized that our strain grows better at 37 than at 25. So maybe a potential human pathogens because prefer our body temperature. So we didn't want to continue further the experiments within. And finally, we have chosen two different two isolates from two species within the Hertzianum clade because they were very rapidly grown at ambient temperature and at 5 centigrades as well. So why this low temperature growth possibility is important? Because in Hungary, maybe here in Italy as well, the pruning of the grapevines happens during the winter or early springtime when the temperature is very low, and we would like to use it as a pruning wound protector as well. So it was important, but further advantages that, for example, Botrytis cinerea has similar temperature preferences, so it's potentially very useful to restrict this disease as well, which is a very important one. So with the help of the university, they provide a proof of concept support. They provided the registration of these strains. So the patent, and it costs as well a lot, and you risk, maybe you patent it, but it's not marketed. And they also helped us to find a company for the final registration for usage, which is a must, and it was the Danuba, and they got the registration in Hungary at 2022 at May, and from this time they are on the market. They are on the Hungarian market, now they are in the Slovenian market, and we are about to get the permission to be used in Portugal as well. So possibility maybe in Italy, but it depends on each country about the needs for the registration. So just step by step. So those strains are really endophytic ones because we could re-isolate them from the woody tissues following one single treatment. We treated soaking the young plants cut before planting, And 5 months later, we could re-isolate so above this soaking level. And we could identify them with the molecular markers that those were our strains. We could detect their colonization on several different grapevine plants, not only from white varieties. The full mint is a white variety from Tokaj vine region, but from international red varieties as well. We could detect their effect and re-isolate. Because the plantation, when they were originated, those strains, so the vineyard, and the vineyard for the first trial, when we could re-isolate 5 months later, were not ecological, so they used a lot of pesticides. Then we were interested about it, whether they can tolerate those pesticides, some of the fungicides or not. And at the time, the fungicides, the systematic which can enter inside the plants were used. We tested, none of them could completely inhibit the growth. Okay, so some restricted the growth of our strain but didn't kill them. So they adapted maybe for these pesticide usages in the farms. And finally, I would like to show you some of the results we had. First of all, the first step, okay, they were isolated from plants, maybe from healthy plants. So let's test their activity in vitro in the laboratory. The most easiest test and the first we do, it's a confrontational test when we inoculate on the same plate the pathogen or the trichoderma. Let's see how they can restrict the growth of the pathogens. So sometimes we have to give 3 days for the pathogens start to grow, and we inoculate later the trichoderma because they can grow very fastly. Okay, just leave some space and for the pathogens as well. Huge majority of the pathogens were completely overgrown within 3 or 5 days by the trichodermas. And not only the ones isolated from grapevine, and pathogens are participated in the grapevine trunk diseases, but also some pathogens which can cause woody diseases from other plants, and they were isolated from walnut, so they could repress. Moreover, we had at the laboratory some oomycetes, so not real fungi, and they also repressed their growth. So 100 biocontrol index means that the trichoderma completely overgrow, just you can see on the slide, the pathogen, because it's green, and started to sporulate on it. And sometimes we could detect this kind of mycoparasitic activity as well. So we could detect and prove that they are effective against plant pathogens, several plant pathogens. And we also could detect their biostimulant effect. So we were soaking before planting the plants, the grapevines, in spore solution of trichoderma, and we planted the plants. And even 2 years after the only treatment, we could detect their significant increase in bud burst ratio and in bud burst vigor. So they developed, the buds developed better. Okay, that was the only we could calculate, because we tried to measure the shoot length, but it was too complicated. So it was visible that they grow better, but it was difficult to make a measurement and the statistical analysis for it. Okay, so this effect on biostimulant was reported last year when they used the product itself in 5 hectares, so not only in some plants. And they were soaking. You can see the soaking method, so it's very simple, before planting. And at that year, it was a drought, in severe drought in Hungary last year. So after the planting, there was some rain, but from the middle of April till the end of June, no precipitation at all, nothing, okay? And it's at September, the owner were very happy because none of the planted grapevines died, okay? And within 1 year, 2.5 meters long as a growth. The only problem that they have started to produce some grapes. So for this year, he had to buy all the harvesting equipment as well. So, okay, but finally he was very happy to use this product. So this biostimulation, we also can sometimes, or not we, independent research, because it's within good experimental practices. Okay, these research were, they could detect the increase in different ratio, the yield, as well as the increasing the sugar content. Okay, so the quality of the grapes were increased as well. We also tested the biostimulation not only on grape, but on crops. First time I thought, okay, they will not work. So maybe on other fruits, maybe they will work, but on crops, I don't think so. Okay, but we had some preliminary results and we started again in field. So it's very important that whether these strains and the product can be applied and has a good results only in the laboratory, in greenhouse, or in the field as well, when there are a lot of other microbes, different environmental, not always controlled environmental conditions. But to test them on the field has some problems. For example, if there is not a homogeneous conditions, otherwise, then the results may not be good, okay? So we had this field when there were preliminary results, NDVI results 5 years back, and we could exclude from the analysis a small part of the field when the NDVI showed that there was some restriction for the plant growth anyway, okay? And we had a repetition and we collected, the harvest happened with a precision combined equipment. Okay, so from each parcel, they get the data about the amount of the grains, of the maize grains, okay, the yield. So we had several repetitions and we precisely knew from which part they were originated from. Okay, so there was one treatment when the maize was between 5 and 6 leaves, okay, as a development. And after the treatment, 2 weeks after the treatment, we could detect significant increase in the number of the green leaves. So this year was a drought. So majority in our region, there were no yield at all for the maize. For this parcel, they had they could harvested some maize, but when they used trichoderma, because more green leaves, so protected a little bit from the drought, higher spad index, maybe you have heard it can measure the chlorophyll content, so the photosynthetic activity. So it was also significantly higher for the treated plants and almost doubled the yield in drought conditions for maize. So it was something we were very happy with. Because of the drought, there were no diseases, so we couldn't detect the biocontrol effect. Some other field effects, so it's small parcels, it's a good agricultural practices in Merlot, it's a grapevine, you know, it's very widespread, international grapevine. With increasing the dosage of the product of the trichoimmune, we could decrease the downy mildew symptoms, both on the leaves and on the branch. And at the same time, we could detect higher yield and again, higher spad index when they measure. In Slovenia, we tested on resistant cultivars Okay, there was a difference, sometimes not very high disease incidence, for example, botrytis cinerea, but it could be decreased with usage, with combination of chitosan, several usage, and oidium as well. And a strange disease I met only there, it's anthracnose. They detected it was very effective in this case. It seems to be the efficiency in this case. In Phytophthora infestans, we tested on potato because it's a devastating disease and you have to use a lot of pesticides to maintain in a or to protect your product from it. There was used only 2 times the trichoimmune, the product, and they could decrease the infection rate and increase the yield again, okay? And we couldn't measure, but it was reported from producers that it was very effective against Fusarium. Fusarium disease in the after tubers. And Fusarium blight was also decreased for the wheat. And again, when we increased the dosage, then there was a significantly decrease in the disease incidence and disease severity. And we tried to detect the yield increase just a little bit, so it was not significant, and the toxin production, but it was not very high. It's, however, we think we will try to do some more experiments or order independent experiments for the registration, because we think because they can restrict the Fusarium, they can restrict the growth of Aspergillus, they will be able, with the usage of their, we will be able to decrease the mycotoxin content of the grains as well. Some others, for some flowers, for the foliar diseases, we could also decrease, and efficacy was quite nice in these cases. And just some more, 2 more fruits. In case of apple, we have several checkings. One of them also proved that against oidium, it can be effective as well. In case of Venturia, you have to apply it several times in the high dosage, but some of the eco producers, ecological farms, they can use it effectively and against Monilia, but we cannot get always the numbers for it, okay, for the data. And with several times we can detect the increase of yields, increase of fruit weights, increase of leaf spotting index, increase of some other active ingredients. And in plant, in stigmina, which makes a hole in the leaves and decreasing the photosynthetic surface, they could also decrease these symptoms and increase again the yield, the fruit weight, and the spot index. So as a conclusion, there are several advantages of using those endophytic strains which are isolated from inside the plant and from agronomic crops, that they have adapted to agricultural practices, so they can tolerate a little bit the fungicide usage as well, so they can be integrated also in IPM, integrated pest management. So in this case, you also able to decrease the pesticide usage. Not the only way is to apply them in an ecological farms, and they adapted to Hungarian climate conditions, because the other strains from France with a lot of precipitations, from the States, from different conditions. So, and they are eco-friendly because they're not foreign ones, okay? They are widespread in Hungary, but those species are very widespread in Europe anyway. It was reported. They adapted and they tolerate the low temperature as well, which is very important. They have adapted to host and its microbiome. We have never ever detected the toxicity for the plants, okay? And the plants are healthy. So that's why they are acting just like a probiotic, okay? They cover the space from those pathogens. They can enhance the immune activity of the plants and have several beneficial effects, not only to repress the pathogens, lower the risk for soil microbiome because they have a tendency to invade the plant itself, okay? And they do not kill, really, all the other microbes. They have not restricted any of the tested bacteria either. And pesticide application or antagonists do not decrease their activity after they have colonized the plant because they are inside the plant and the majority of the pesticides just act at the surface, okay? And the other microbes can survive in the surface. So we have published some of our results. We have a patented product on the Hungarian market, which is available and use several conditions. So thank you for your attention.
Thank you. A lot of items for discussion. Maybe something that is shaping your curiosity, even in Italian. Better if in English. Even if in Italian language. One question each of you. Who wants to start?
I want to know what is the acceptance from the agricultures to the use of this kind of bio.
So it's a very nice question because we are facing that doubt. Okay, sometimes they have used some products, but they couldn't see the difference. We also have to ask them just to try to trade some parts and do not use on other parts and have a look at by themselves the difference. And the other problem is that they previously used some, which sometimes they also check didn't contain any living microbes. So you have to take care about the carriage, about the keeping of these because they are living ones. So, and they also have to take care about the application, the timing of application. So we also try to optimize them. And I like the company because they not only selling the product, but they educate the ones who are using because it is a must. If you are using some biological product, you have to educate the people because without the knowledge, you will not see the effect and about the combinations, not to use at the same time with the copper because maybe they will kill them. So it needs a lot of education. So for the producer, for the farmers, they need an open mind and there is a huge restriction as well and knowledge they have to learn. One question. One question.
Not understand, but you have to give trichoderma only one time.
It always depends on your aim. So if you want to use as a biopromoting to promote the growth, you can use only one time and you can see the effect towards several reasons. Or if you want just to protect from the pathogens which may affect during the plantation, the early time, it's okay to use only one time. If you want to protect from a microbe which is coming outside from bacteria, oidium, and maintain only at the surface of a plant, you have to apply several times. At the same time, when you would apply some pesticides as well. But for example, against GTD pathogens which are living inside, okay, you can apply once and they have a preventive effect. But if you prune them, again, a gate, just at least protect the gate from the new invaders. So they are not miracles. They can help, but they will not overgrow. Just like the number of an army, okay? They can protect, but if there are a lot invading, you need some more. So for the horticulture, for example, for apples, we used it during the plantation. And after a year, even we couldn't realize that it was not planted in the previous year.
No.
Okay, so it increased the growth, so effectively. But okay, now it's in the market. So when they applied by the users, I sometimes got some reports because it's for home and garden as well. And sometimes they ask me, how can I apply? Could you give me some advice? And they report, oh, just have a look at my roses. They have several flowers. They are wonderful, but we cannot count them, okay? That was the first time my father-in-law didn't suffer the monilia and we could collect the cherries from the tree. Oh, that's great, but we cannot count them. So we have several this kind of positive effects. Some of them, okay, it was a frost, so I will not use because it's not useful. Nothing can help our plants, or okay, I couldn't realize the effect, the differences. Sometimes it was because the weather was so hot, and above 30 centigrades, it cannot work and to save the plants from the pathogens. So we need more data, but we could realize several positive effects.
And the questions are open also for the people that are following online. So just feel free to open the microphone and take the question. Somebody else? Okay. Somebody at home wants to ask something about the first or the second talk? If not, we thank you so much because it was very kind to share with us your knowledge and the options about the University of Debrecen and about the opportunities of network. And also some very interesting data about this trichoderma that I'm confident will become pretty popular. And maybe one day you will be technician in the field and you will tell, oh, wow, this was the trichoderma that Professor Carafa told us. Yeah, it works very well. I will use in my farm. It takes time, it's not so easy, but the direction toward those new compounds that works with a different mechanism of action. So there is an induction of defense in the plants, and the induction of defense is very popular. It's becoming more and more popular. This linked to an action direct toward the pathogen. So this is double mechanism of action that is very important. So you talked about also some production of OX, volatile organic compounds. So which are and how many mechanisms of action this trichoderma can have toward the different pathogens? Which is your opinion about, if you investigate or you want to know more?
We would like to know more and doing some studies about even the molecular gene expression increasing, studies as well, because we were just about to measure the chlorophyll content by SPED and by chemical way of action. So there were reports. We would like to prove that our strains also are able to increase the expression of the genes responsible for the yasmonic acid, for example, way, or in important in drought tolerance, so in immune and drought tolerance as well. And for the photosynthesis activity, how they do change, and maybe about because we could detect the difference between some polyphenols and antioxidant contents of in case of some horticultural crops and fruits. We would like to study this way of action as well. So it seems to be that both the chemicals and the plant extracts and some of the microbes act in similar way, and they can help each other. For example, we have tested together, in field, they apply together with algal extract, and they work very nice together, as well as they are not inhibiting the growth and the activity of mycorrhiza. There are some publications and we have the same results, okay? So although mycorrhiza are fungi, trichoderma will not attack them. And we do not know the mechanism why they do not inhibit the fungi, which are helpful for the plants, okay? So they seem to be selecting. So they kill the pathogens, the bad ones, but they do not inhibit the good ones. It seems to be. There are publications, so we would like to do some more research on this field.
Okay, thank you. Any other question? Okay, so thank you so much, and let's think on those issues because they will be part of the course that we will follow, and let's keep in touch for further interaction. Thanks to you, and thanks to the people that followed online, and let's go forward towards sustainability. Okay. Thank you. Thank you.